Okay, so I'm going to quickly review DNA replication and protein synthesis for you. We're going to start out talking about a nucleotide. This is kind of where it all begins. We start talking about this in our very first day of class when I introduced the monomers of nucleic acid. And these nucleotides are very important because they make up all of our genes and they make up our DNA and our RNA inside of our cells. So we have 5 different nucleotides. We have adenine. That is, we just use the A. Okay, and it's a molecule that has 2 carbon rings. So it's kind of like this little shape here. It looks like this. And then there's one that looks a lot like it. That is guanine. Okay, and so it kind of has this 2 carbon ring shape. And we label these guanines with a G, right? And then we have thymine, which is just a single carbon ring that we label with a T. We have cytosine, which is a single carbon ring. And remember, these have different constituents, like different molecules coming off them. So this adenine has a nitrogen coming off up here, and this one, guanine, has a nitrogen coming off down here. So these little bins are carbons, but the rest of these things that are on the outside, whether it be oxygen or hydrogen, are different, and that's how these differ. Okay, so thymine has an oxygen that comes off up here, where cytosine has a nitrogen that comes off up here. And then we have one more, uracil, that is also just a single carbon ring, and we only find this one in our RNA. The rest of these are found in DNA. So this is kind of leads us to our next question. And so I guess if we're going to say what are nucleotides, these are molecules that make up DNA and RNA. Okay, so how do these nucleotides and RNA and DNA differ? Well, in RNA, the C's go with the G's, and the G's go with C's, and then the A's go with the U's on a strand of RNA. And then in DNA, the C's still go with U's, C's still go with G's, and the A's go with T's. And this is how you're going to tell if a strand is DNA or RNA, because it'll have something like CT, A, C, C, G, A, T, T, and we would know that this was DNA. Okay, of course it would have another corresponding strand, and we'd know if something was RNA because it had U's in it. U, C, U, G, G, A. Because of those U's, we would know this was RNA. Okay, what other differences does DNA and RNA have? Well, DNA is single-stranded. Double-stranded. Double-stranded. And RNA is single-stranded. Which means when we say strand, we're talking about that backbone of DNA that we learned that deals with that phosphate group, that ribose sugar, and that nucleotide. And then here's our nucleotide. So it's either a carbon ring, so if it looks like this, maybe it's thymine here. But this is the backbone that is what we're referring to for the strand. So this thing builds and you have a whole bunch of them, right? And say this is a C. Okay, and this would be RNA because there's only one side. Now if there was another side over here with another phosphate and another sugar and then a hydrogen bonded to an A with a phosphate, this would be double-stranded because it had two backbones. Okay? So what process doubles the amount of DNA? Well, this is what we learned about, and this is called DNA replication. Okay, and so DNA replication is doubling the amount of DNA in the nucleus. Okay, and this is how our cells replicate. So we learned about DNA replication first when we were talking about the cell cycle. And this one says right here, what phase of the cell cycle does this occur in? This is the S phase. Okay, so when we are talking about the cell cycle here, we have our G1, our S phase, G2, and then the M phase. Okay, this is where DNA replication takes place, right here. And this is very important because if you're going to make two new cells, right, here's our two little cells, they both have to have a full set of DNA in order to function properly. And so DNA replication, it's important because every cell needs a full set of DNA. So it has to be replicated. Okay. All right, how does this process work? All right, so let's get our drawing out here. I'll change to red. Okay, so we're going to have our strand of DNA. We're going to have a little replication bubble, and then we'll probably have more replication bubbles, right? We'll have a couple of these things occurring. So here's our other side of our DNA. We have our replication bubble, other side of DNA, and this happens all over. Okay, so this is our DNA. So this is going to have A's and T's and C's with G's and T's with A's. Okay, so A's again going with our T's and C's going with our G's and vice versa. And then right here, this is the place we call the replication fork, where this is going to start splitting. And the molecule that splits that is called helicase. So if we have a molecule right here, this is called helicase. And this is an enzyme. We know that because of the ASE ending. And its job is to unzip the DNA. Okay, so this is why our DNA starts opening up here, and we're able to see what nucleotides are there so that our other enzyme that's important in this, which is DNA polymerase, can see what nucleotide it needs to find in the nucleus floating around, and then it can put it into our new strand of DNA. We'll get orange here. Okay, so we have one that, if we say this is a 5' on this side, this is 3' down here, this is 5' here, and this is 3'. Our DNA polymerase always runs from 5' to 3', and that one is always going to be called the leading strand. So this is our DNA polymerase here, and he's going to be moving in this direction. And behind him, he's going to be making a new piece of DNA. He's finding all the matches. Okay? And then we're going to have another one moving from 5' to 3'. That's the way enzymes run on DNA. Okay? Moving this way, and so it's building DNA backwards. Okay, so here's its DNA that's building. And this one's a little harder. It's going to build little pieces of DNA, and then we call those Okazaki fragments, and then we have lysase that comes in and glues them together. And it's a little more complicated on our lagging strand. It takes a little longer, but that's the way the enzyme has to run. And so this one is called our leading strand. And this is going to occur on all of these little places called the replication bubble. And so what we get at the end is we get semi-conservative strands of DNA. And the reason we call it semi-conservative is because we have one new strand, new backbone, new nucleotide, and we have the red one, which is our old strand, right here, the template. Old strand, or we can call it the template. Okay, and so we call this new DNA strand semi-conservative because it's half new and it is half old. Conservative, and then it's half new and half old. Okay? All right, we talked about helicase, 2 DNA polymerase. I should label this DNA polymerase. Okay, this is all happening in the S phase. We're going to get double the amount of DNA. That's going to make our chromosomes for our next stage. Okay, so let's go to this page. So key vocabulary, definitely know what DNA polymerase is. Need to know that, and it's going to build and edit DNA. Need to know what helicase is, and it's going to unzip DNA. Okay. Replication bubble, and that's where that is actually occurring. Replication fork. Replication bubble is where replication takes place. And then replication fork is where DNA is unzipped. Replication starts. Replication starts. Okay, so it says, what's the other process that occurs in the nucleus? Well, we've actually learned about 2. We have DNA packaging, but that happens during prophase. Oops. Packaging. That's an A, G, packaging. And this happens during prophase that we learned earlier. This happens in the nucleus, where we make the nucleosomes and our DNA wraps around histones. But that's not the one we're talking about. The one that we want to focus on for this is transcription. Okay. So the steps of transcription and how it works is we're going to have our DNA strand again. Okay, but this time we're only going to have one gene of DNA. So we're not looking at the entire strand of DNA. We're just looking at one little piece, segment of DNA. Okay, so DNA is double-stranded. Has the A's and the T's and the C's and the G's. And then it's going to have a little ta-ta box here at the beginning that marks the beginning of our gene. We're going to have replication, transcription factors that assemble here. And these factors are kind of like the little flag that says, hey, this is where transcription begins. And then we're going to have an RNA polymerase. So now we have RNA polymerase. And RNA polymerase, its job is to make RNA. Okay, so it's going to run 5' to 3'. It's going to zip down this DNA strand. And out the top, we are going to get a piece of RNA. As it moves down, it sucks in nucleotides, and makes a single-stranded, and this single-stranded piece is our RNA. And so this RNA is going to be the end of transcription. So transcription is literally the process of turning DNA into RNA. Okay, so our first step would be the assembly of the transcription factors on the TATA box. Second step would be where our RNA polymerase attaches. Third step is the RNA polymerase running down the double-stranded DNA, unzipping it, binding our new nucleotides, and making that RNA strand. Okay, so now that we have our RNA strand, we need to edit it. So RNA gets edited inside the nucleus, and this is where we're going to split that RNA piece into exons and introns. Okay, so remember, our RNA is now going to have U's, no T's, and we're just going to take the information that is vital for the protein. And so that might not be all of it. Okay, so we'll say we break it like this. Okay, and these are the exons here. These are the introns. These are the exons. And so this piece right here is going to get spliced out by the spliceosome. And it's actually going to go back up into this RNA polymerase here. And this is where that RNA polymerase is getting its nucleotides, is from the introns. Then the exons are going to come together, so you'd have a new piece of just exons, and that is mRNA. So we'd have the exons, which was A, U, G, C, G, A, U, U, A. And this is now our mRNA. So editing is taking our RNA and turning it into mRNA. All right, so it says what happens after the RNA is made? The mRNA leaves the nucleus. And it goes out and finds our RNA. And this is where translation begins. So where does translation take place? It is in the cytoplasm. And it is at the rRNA, or the other word for rRNA is ribosome. If you remember, the ribosome was really small when we were learning about our organelles, and now it's really, really big. So we have our ribosome here, it has 2 subunits, and our mRNA is going to come and it's going to lay down in this groove here. And we call each of these little 3 sets of nucleotides codons. Okay, so right here, this is our first codon, and it's usually the one that starts, is AUG, our start codon, and it's going to attract an anticodon. The anticodons are carried by the tRNA, and so the tRNA are all over here, little triangles with anticodons right here, and then they also carry that amino acid at the top. So the tRNA that it would attract would be a UAC anticodon. We'd have our triangle here, and then our amino acid would be at the top. Okay, so if this had been running for a little while, we would have a long string of amino acids that we're going to fold in a very particular way, making this polypeptide chain, and these were our peptide bonds, and this is going to produce our protein. Okay, so we'll put our amino acid here. This is our next amino acid, and every time one tRNA leaves, another one comes in. So there's always an empty place that gets filled by a new tRNA until the stop codon. So if this was, let's see, what was it up there? CGA. So the tRNA that's going to come in, the anticodon, is going to be GCU. And then we're going to use this codon right here to code for the chart. And that chart is going to be a list of codons that we can find this right here, and it will tell us what this amino acid is. So we know that AUG on our chart is actually the met amino acid, or the methionine. All right, so almost done here. What is protein synthesis? Protein synthesis consists of transcription, editing, and translation. And so it really is from one gene to one protein. And then how would a mutation affect this protein synthesis? Well, if we have our different types of mutations, maybe we answer this one first. So we have our point mutation, which is a substitution. Of nucleotide. And we have a frame shift mutation that is either going to delete or insert a nucleotide. Or we have our chromosomal mutations that are called gene mutations that are translocated. Okay, and so this is where chromosomes move genes with a mistake, move our genes. Okay, and so like a point mutation, this is going to affect protein synthesis, because if you were to take this code right here and substitute this G for a C, now all of a sudden it's going to change this anticodon, which is going to change this amino acid. And that's a pretty big issue, because now our entire protein is going to be non-functioning because it didn't start with that correct first amino acid. And so a substitution is going to render the entire protein not to work. Okay, frame shifting is even worse. Okay, so if you were to come up here and take this one out and move this whole segment down, now we're really going to have an issue, because now we're going to be lacking a nucleotide at the end here, and every one of them is severely changed. So now all of this information gets shifted down and every amino acid is incorrect, which is definitely going to give us a non-functioning protein. Same if one was inserted. If we added an extra A in here, that's going to cause an issue, because it's going to move everything down this way. It's going to shift that entire frame, as the name implies. Okay, so it's going to make a protein that doesn't work. Okay, and then how are viruses related to this information with protein synthesis and DNA replication? Well, viruses are going to attack our cells. We have different types of viruses, and basically they are a protein shell, and they have nucleic acid inside. This nucleic acid could be RNA, it could be DNA. Sometimes they even carry their own enzymes for replication. But the reason why we are doing a virus project is so that you guys can understand how these viruses are replicating inside of your cells by using your information. So whether it be if they come in with RNA into your cell, some are taken in by endocytosis, like retroviruses. And then sometimes, like a phage virus, will actually just put its DNA right into your cell. Kind of just push the DNA in, or it might put in the entire thing, the entire protein capsule. And then that DNA goes into the nucleus, and it either can insert itself into your DNA and replicate to all of your cells. It can be just RNA and use your ribosomes. So it might just hijack your translation. It might hijack your transcription. Or it might hijack your DNA replication, your transcription, and your translation. So these viruses cannot survive without you. They cannot replicate without you. And they definitely can't spread without some kind of living vector, a living cell that they can hijack these three processes that we learned about in this unit. And that's the only way they can survive. So can only survive with you. Okay, so hopefully that is helpful. We are in Chapter 8. If you wanted to refer to your textbook, we did sections 8.3 on page 221, 8.4, which is 225, and 8.5 on page 229. We also did some of the mutation stuff in section 8.7 that starts on 238. So in your textbooks, it would be pages 221 through 239. 221 through 239. If you'd like to read about any of this information before your test. All right, have a great night.